is the originator of it for a reason that he had a very bad accident 8 years ago?
Almost 10.
Almost 10. And James is the scientific brains behind it. So first of all, I'm going to ask you, Matthew.
James is the brains behind it, not just the pretty face.
Okay. I'm going to ask you, Matthew, first of all, why it's so important to you.
Okay, thank you for the question.
Actually, and the basic question, what is it?
I know there's no time to squander here, but I would be remiss if I didn't at least start by thanking Mark Anderson for his uncommon kindness and generosity, and the rest of his team, Sharon, Sally, and Shane, for their patience and tolerance in working with me, which is not always easy, and they've been great.
Okay.
So, and now, if you don't mind, I'll rephrase your question so that I can ask my own question and then answer it, which is what I'm better at.
That's absolutely fine.
So, I think the question is, what is NeuroRepair, what does it do, and why should you care? NeuroRepair is, as Steve has already stated, a company that I founded after I had suffered a neurological injury in a sports accident. Absent that, I'm sure I never would have even thought of the idea of neurological injury, and it never would have mattered to me. After a very long stay in the hospital, I started to do some research, looking for someone who was doing something in the nature of neurological repair. This was after months of hearing one doctor after another telling me, in virtually these exact words, brain injuries never heal. There is no treatment. You will never recover. It is a bedrock precept of medical science that once you lose neurons, that's it, you're done. They're never replaced, they never heal. You're just not going to get better. And having done some research, looking all over the world for someone doing something, I discovered Jim, who was geographically convenient to me, I being in San Diego, he in Irvine. And also, he was one of just a handful of people working on this very important question anywhere in the world. The reason for that is, of course, if you go to medical school or you get your PhD in neuroscience, and your Nobel laureate professor is telling you over and over, brain injuries never heal, there's no such thing as neurogenesis. Once you lose neurons, they're gone forever. They never heal, they never repair, there's nothing to be done about it. So don't bother. Don't even bother working on this. And virtually nobody does. But Jim did. He's stubborn that way.
I'm watching the clock here, so I'm going to turn to Jim now on your excellent cue and ask Jim where the science now stands. I should say that Matthew, 8 weeks earlier, before this accident, had sold his company. A chap purporting to be Bill Gates phoned him up and said, "I'm Bill Gates, I want to buy your company." And Matthew assumed he wasn't Bill Gates, but he was Bill Gates.
I really responded.
And he did buy.
Who is this really and what do you want?
It was Bill Gates, and he bought the company, and that money is what's funding this research, which is very exciting research. And Jim will tell you why.
I had thought when Matthew contacted me, it was because of the quality of my work and that he had heard that I was willing to go against some of the doctrines of neuroscience. And it's a previous work with Parkinson's disease. And now I found out he just called me because I look the most like Mark Anderson.
Both of you should sue.
And this would be a little bit of death by PowerPoint. I have to show it because it's anatomy and it's hard to explain. It's easier to just look at. So we look at the first slide. And here are a couple of the doctrines, as Matthew so well said. First of all, that brain damage is irreversible. And part of this is that the idea that there are very few stem cells in the adult brain. And the ones that are there were always thought to be simply there to cause scar tissue. That is, it was glial cells that would be created and would cause scar tissue. And in fact, these stem cells that were in the brain are probably one of the main reasons why things like embryonic stem cell transplants, engineered cell transplants, which we've done, do not work because these come in and there's a reaction set up against these cells and the scar tissue forms. And so we had started to work with growth factors. As a point, our lab was the first to discover a growth factor in the mammalian brain. That was back in 1984. Now, with all these years going by, you figure, haven't you done anything yet? And it was kind of a long way to go. We really had no idea because nobody else was doing this, and it took us a while to then look at a bunch of other growth factors. And one of them was the sister compound to this epidermal growth factor called transforming growth factor alpha. And we had started to look at it using the doctrines of the day, back in the, the 80s and 90s too, which is you had to do cell transplants. We had the cocktail with these different growth factors. And then, as it turns out, most of the stuff by serendipity, a student made 2 errors. Forgot to add the cells, just put in the growth factor, and did it in the wrong place at the wrong time, and voila, instead of seeing a couple of new cells, we saw hundreds of thousands to millions of new cells, which we fell off our chair. That was doing Parkinson's research, our main interest. Now, then Matthew called me after our study had gone from NIH to Congress, to the stem cell report to Congress, saying, oh, maybe adult stem cells, that is endogenous adult stem cells, might be able to be mobilized in great numbers and directed toward an injury if you do it right. If you put it in the right place, the right time, and all of that. So he called me up and I said the same thing the doctor said. I said, this is impossible. I can't take the young students and postdocs in my lab and have them risk their careers for several years going after something we know doesn't work. He convinced me of it. I don't know how. And I do.
I said I had some money. That was it.
Well, there were a couple of bucks there. Well, that doesn't hurt, Sure, for some characters.
It was naive on my part, but it worked.
And so anyway, yeah, he was willing to fund it, and we went ahead, and so here's what we found. So if you look at the next slide, I only have 3 slides. The second one, at the top left, you see a human stroke. And this is the most common stroke, and in fact, it's from the artery, that's called the artery stroke, and it's in the basal ganglia and partially cortex. We take animal models of different diseases. In this case, we do a stroke model in rodents, and that's at the top right. And it's the same sort of picture here, stained with the stain that shows new cells being made over in the top right, and they're migrating. So there's millions of new cells, and they migrate right to where the injury is and also the growth factor is. You have to have 2 together. If you do a Venn diagram, it has to be overlap and it has to be injury. Otherwise, you get this temporary effect and it goes away. It's a key deal. And so, in the bottom 2 pictures show as if you're sitting in the ventricle, because the adult stem cells are lining the ventricle. So if you're sitting in the fluid of the ventricle, those bottom 2 pictures are the actual cells, stem cells dividing. So that red one there at the surface is a stem cell, and at the right it's starting to divide, and then it migrates. So in the last slide, what we have here is we did it a couple of ways. When we tried doing this, first of all, next slide, we tried, and this is an animation, and this shows that area of the stroke, in the common area of the stroke. You see that cell lit up because it died. And the idea is you have this circuitry in the brain whereby what you're thinking, your will, coming from cortex, directs this basal ganglia where these automated motor and sensory programs are, and then that forms loops, and that tells your body what to do. In the case of stroke, that's gone, so you know what you want to do, but you can't do it. And the idea is you have to restructure the brain. And so one way to do this is you try to do all this engineering of cells. The other thing is just to take the latent potential of the brain with a natural growth factor, TGF-alpha, which is a native growth factor, and you put it in there, and voila, as you can see on the right there, those stem cells start to divide like crazy, and they migrate right to where this 5-alarm fire was. And so we've done this in a couple of models, and it worked, and none of the people in our lab could believe it. And this led to a 98% improvement of the behavior.
So, because I'm watching the clock here, where you now are is, it's worked on rats,
right,
and may work on humans, and you are talking to the Parkinson's Society in Britain about collaboration on a further trial. Am I right?
That's correct. We've just had some preliminary conversations, by the way, set up by Mark Anderson, with Parkinson's UK. I got an email this morning from the person who's been helping us with them, saying that the Stroke Association of the UK is also interested in participating along with us. So this could be a perfect fit. Those two organizations are very keen to have some sort of clinical trial. Everyone, of course, is very anxious to make some progress on these two really terrible maladies, Parkinson's and stroke. They have the facilities and they have the money. They don't have the IP. We have the IP, we don't have the facilities or the money.
Right.
It's a very nice marriage if we can make it work.
Absolutely. Good.
Can I make one more point?
You can.
The key here is this is not an acute stroke. This is an old stroke. That's what really is remarkable, that this is a stroke that's equivalent to a 1 to 2 year old stroke in a human. It's not acute stroke. That's what really blew us away. That's all I needed.
Good. Thank you both very much. Per.
Yep.
Tell me about your company and well, don't tell me.
Okay.
These are the important people. Tell them about your company and where it's going.
Yeah, we're changing.